This video is about stage 4 of metabolism, the electron transport chain, and oxidative phosphorylation. So, stage 4 really involves taking the reduced form of coenzymes, and these are from stage 3 of metabolism, the citric acid cycle. They're from stage 3 in citric acid cycle, and in the electron transport chain and oxidative phosphorylation, we really convert these to energy in the form of ATP. And let's be honest, we eat food for energy. And so we have to, we want to learn more about how it is that that energy is formed. So let's look at a detailed view. In electron transport, the reduced forms of the enzymes NADH and FADH2 are converted to NADH plus into FAD, and they give up 2 protons each, so 2 H plus each and 2 electrons each. At the same time, oxygen plus 4 protons and 4 electrons reacts to form water and releases energy. So the net equation is NADH plus H plus FADH2 plus O2 reacts to form NAD plus FAD plus 2H2O. Now, oxidative phosphorylation takes energy plus ADP plus a phosphorus, or a phosphate rather, and reacts to form ATP and water. So it's really ADP diphosphate plus another phosphate reacts to form ATP, which is really triphosphate. So the energy that's released from the electron transport chain, the protons, the electrons, that is being used to actually add the phosphate in ADP and make it ATP. So take the diphosphate and make it triphosphate in oxidative phosphorylation. The vast majority of ATP is actually made in stage 4 here in electron transport and oxidative phosphorylation. So a big part of what we're going to learn here about stage 4 electron transport is that we really are setting up a proton gradient, right? They're setting up this excess of H pluses, and that excess of H plus that's produced really is what drives energy production in stage 4 to be able to produce ATP. So it's really this proton gradient, or all these protons being produced, that's really kind of the engine driving this whole thing forward. So some big picture ideas about electron transport. It's really a chain, a sequence of reactions, or a chain of events. It involves 4 enzyme systems. Electrons are going to get passed from one complex to the next to the next. This actually happens in the inner membrane of the mitochondria. So you may have heard that the mitochondria is the powerhouse of the cell. Well, this is where those, transformative energy creation reactions are actually taking place. When we say powerhouse, we mean really in that inner membrane. It starts with those reduced forms of NADH and FADH2 that we get from stage 3, the citric acid cycle. So the NADH and the FADH2 from stage 3 in the citric acid cycle. They're really electron rich. And then in oxidative phosphorylation, sort of the next part of it, the H plus, or the protons generated by electron transport, those proton gradients really drive ATP production. Remember when I was saying it's when we have this excess, these H pluses are our proton gradient. That's what will drive ATP production forward. And so it's kind of fun to actually take a look at what this membrane, inner membrane of the mitochondria actually looks like, just to get a little bit better idea of what is going on with electron transport. The big picture ideas are that we have from NADH, and from we have this sort of, chain of electrons traveling, we said, through four, four separate or different enzyme systems. So we've got these four enzyme systems, and these electrons are going to get passed through these four enzyme systems. NADH and FADH2, they're actually getting oxidized. Let's go back to that actually for just a second. If we go back to like the net reaction here, in the net reaction here, NADH and FADH2 are being oxidized by oxygen. The oxygen's actually going to get reduced. These are actually going to go, these coenzymes, NADH and FADH2, they're going to actually go back to their oxidized forms, NAD plus and FAD, and oxygen itself is being reduced. Okay, so they're being oxidized. As they're being oxidized, as NADH and FADH2 are being oxidized, they're producing these 2 protons each, and these 2 electrons each. So if we go back to this mitochondrial membrane, what we can see is we've got, this we've got cytoplasm on the outside, then we've got this outer mitochondrial membrane, we've got this bilayer we can see of heads and tails, with the tails together forming that outer mitochondrial membrane. In the inner membrane space, we've got all these protons, all these little H pluses. Then we've got an inner mitochondrial membrane, so another bilayer where we've got tails, nonpolar tails facing each other and polar heads at the outsides of the membranes. But in this membrane, what we actually see are that, we can start to see, those 4 different enzyme systems that we've got in here. They're kind of labeled just 1, 2, 3, and 4 in the different enzyme systems, but they're in the inner mitochondrial membrane. Okay, and then we see in the inside here, we've got labeled NADH, and we're going to have these electrons, traveling through this series of these 4 enzymes, and NADH gets oxidized to NAD. FADH2 is going to get oxidized as well. And as that happens, like we can see even from this first enzyme system, protons get generated. And so as these are occurring here, we see across, system 3, protons generated. Across enzyme system 4, protons are generated, and they're passing from inside the mitochondria to that inner membrane space, until that inner membrane space has this gradient, or these excess H pluses. Now it's those excess H pluses that can pass back through the inner mitochondrial membrane through ATP synthase, through this proton channel in ATP synthase. Because there's this excess in that inner membrane space, they want to pass back on through the inner mitochondrial membrane, into, the center, essentially, or the main part of the mitochondria. And so as those protons go from that inner membrane space through ATP synthase, ADP is actually converted to ATP with that proton transfer. So those protons that were generated, all these H pluses, so all these H pluses, again, they've been generated by electron transport. H plus, it's generated, by that electron transport chain. Those are the proton gradient that drives what we call that oxidative phosphorylation, right? They're actually driving, going through ATP synthase so that ADP is converted to ATP, that oxidative phosphorylation, or ADP picking up the P, the extra phosphate group, to become ATP, to become triphosphate. So how does diphosphate become triphosphate? Well, as these protons are traveling through this ATP synthase to try to balance out this big imbalance, or this gradient of protons. But that proton gradient is really fascinating and a really big drive to being able to form that ATP or that energy. Um, so I just thought it's fun to look into this in a little more depth. And if you are interested in this, biochemistry might be a field that would really interest you because it's a fascinating topic where you can get into all of these details. It's really where chemistry meets this cell biology, and it's really super cool. So, again, electron transport chain and oxidative phosphorylation really happens in this inner mitochondrial membrane in this powerhouse of the cell.